Hello, I'm Ann Montgomery, Editor-in-Chief of BioProcess International Magazine. I'm here in Oakland, California this week at the BPI West Conference and Exposition. A number of speakers have agreed to share their insights with us. I hope you enjoy their comments and find them valuable as you approach your work.
Yeah, my name is Gunter Jagschies. I'm the Senior Director of Strategic Customer Relations at GE Healthcare in Uppsala at our BioProcess division. I have 31 years now in GE and the predecessors. I'm a biochemist by background, living in Sweden with my family, and I enjoy what I do all the time. It's a great world, the biopharma world, to be in. Yeah, well, I mean, obviously it's a process where the product flows from its creation, the beginning, to the end with no interruption, which usually means that it's a very efficient process because you basically don't have any hold times and wait times. Another definition, or part of the definition, is that all the installed parts in the process will always be busy, working on the product in different stages, which makes it very efficient, or which should make it very efficient. And all that belongs into the definition. I know that's not a kind of a textbook definition, maybe that should be shorter, but it's the continuous movement of the product and the fact that all parts always are busy. You don't have any waste in the process. It's all taken out. The answer to that will depend a lot on what product you're actually making. Traditionally in biopharma it has been factor 8, for example, a product that is not very stable long-term in the culture environment. So removing it from the cell culture quickly is an advantage to less stable products. But then monoclonals that are really quite stable have also been made there. And I think there the main advantage is the scale of operation. You can essentially produce the same quantity of product in 1/5, for example, of the volume of bioreactor. Reduces your footprint very significantly. You can then dimension the downstream line accordingly. You harvest basically very small aliquots from the bioreactor. So there is an economic advantage, obviously, right? Capital expenditure plays a major role. Then you have at least a potential quality advantage too. You have basically a living organism in steady state, an environment that is less artificial, I guess, than a fed batch operation would be for that cell. And so there is a chance, not proven really for all molecules, but a chance that quality will benefit from it also, will be more robust. And those, I think, are the key opportunities. There are some niche opportunities, like especially on the downstream side, when you have to only make a limited quantity of material, you will typically not utilize the chromatography resins to their lifetime, so you will basically be paying more than you really needed to for just that quantity. And in continuous processing, you can actually minimize the size of the columns and run this more often, a little bit like a hamster in the wheel. And so you run many cycles for that small quantity of product, you use your lifetime better, and that means you have to invest less into the resins for that particular project. However, that's really only for that niche. If you would continuously manufacture, that advantage would go away because you need, basically the same amount of material in a chromatography column to make the same quantity of product. So if you look at a longer term run or continuous long-term manufacturing, that niche advantage would probably go away. Many people also say it has the advantage of higher productivity. And I agree, as long as you put every step in continuous mode. If that is not the case, then I kind of disagree, because it doesn't really matter that much whether one step is extremely productive if the others are not. You kind of don't see the effect on the overall process in that case. But that's kind of a summary of what is discussed as advantages, I think. For example, on the downstream side, there's no recorded case where a GMP process runs continuous. So obviously people didn't have the technology to turn the downstream process continuous. That has changed in the last couple of years. So now we have actually commercialized large-scale skids, and there are 2 or 3 players who offer laboratory solutions. The automation of that way of processing, handling many columns in cycles, and changing and controlling the conditions of operation, that has only in the last 2 or 3 years been commercialized and become available. What we are still lacking is sensor technology that actually follows product quality or product quantity online or inline while the process runs. So in a continuous process, you operate in the dark to a degree. You run the bioreactor for, I don't know, any time between 30 and 60 days maybe. You don't want to take too many samples. It's a risk for sterility of the reactor every time you do that. And on the downstream side, it's basically the same thing. There's a risk for contamination if you keep that open, so you have to be really sure it's a closed process. We don't have inline detectors for whatever the impurity profile, host cell proteins, whatever. That's one of the difficulties where we need more development. And actually, if that was available, that would change the paradigm, because then you could do anything in processing, because you would always know what you're doing. You would always see what happens. I mean, on the upstream side, many people say it's much easier to develop a perfusion process. I know probably not enough to confirm that out of my own experience, but I've heard that so many times, I just simply take that as a fact now. The difficulties are when something happens, to actually go down, scale this down, have these experiments verified in small scale, and find the issue, do a root cause analysis. That can be pretty tricky, I guess, with a process that runs over a long period of time uninterrupted. And with the sensor technology not really being available, so you would have to go in and analyze this. So you need that expertise in a company to be able to move there. The other issue that we meet most often, that's almost a managerial non-tech issue. The concern that the risk profile of a continuous process is worse than for a batch process. One of the reasons is contamination risk, or lack of control also to a degree. And so right now I describe this usually as, being two religions. Do you want to discuss your religion with someone? No, really it won't lead anywhere, right? People are shooting at each other in the worst case, and we don't want that in our industry, obviously. So there are really different opinions about this, views, also backgrounds, like companies who have already installed facilities, they would have to change things there that may be really inappropriate or just not enough advantageous in a way. So these are often managerial hurdles, type of risk assessment, risk management hurdles. Some people blame the FDA or the regulators for all this because they are conservative about changes, but in fact the FDA management, for example, has really encouraged continuous manufacturing. So at worst, you would have to take their word for it, right? They have to tell their inspectors also, not just speak at management level at the FDA about this. So I think it's often managerial. The technical difficulties can be overcome. We are very optimistic there. But still, we think that it's going to take a decade, or I think we're going to take a decade before we see this becoming more of a mainstream approach, even in downstream processing. Everyone's testing it. Most people get good results, but the implementation is a different story. The manufacturing head has to like it as well, and that's not always as easy. Of course, we need to get more efficient. We need to reduce costs. We need to, make the drugs more affordable. Everyone is in that game. But I think we have, in some quarters, unrealistic expectations on how much continuous processing can really do there. In particular, when that is combined with that risk averseness. You either do it or you don't, kind of, as I tried to say in my talk. Are you in or are you out? Because doing one step is often just a very small improvement that doesn't really count in the billion-dollar revenue world, or hopefully also nice profit levels. And so on the economical side of the benefit list, I would say, apart from the small footprint, the small facilities, which definitely are cheaper, should really offer an opportunity economically. One should not overestimate the economical benefits. It's a different way of running things. In a smaller environment, you can basically put several lines into a facility as it would look now, a building that there is, of the size that typically is used for facilities. It gives you flexibility, but don't think that you can just save on everything. That's not going to happen, I don't think.